Here, and welcome to this episode on bacteria. Back in Chapter 6, when we learned about cells, we briefly talked about prokaryotes. You know, those small, simple cells that don't have a nucleus or other membrane-bound organelles, otherwise known as bacteria. In this episode, we'll review the external and internal structure of bacteria and also discuss some mechanisms where bacteria can undergo genetic recombination to have variation within a population. So let's get started. Because they're small and simple, prokaryotes are believed to be the first cells, and eukaryotic cells eventually evolved from them. They're one-celled organisms and can take a variety of shapes. Over on the left, these spherical-shaped bacteria are called cocci. The yellow rod-shaped ones in the middle are called bacilli, and this corkscrew-shaped one on the right is called a spirochete. Please know that there's no need to memorize the names of these shapes. This is just for informational purposes only. First, let's go over the external features of a bacterium. While all cells have a cell membrane, bacteria also have a cell wall. Like plant cell walls that we've learned about earlier, it helps support and protect the bacteria cell. However, unlike plant cell walls, they're not made of cellulose. Instead, they're made of a protein-carbohydrate complex called peptidoglycan. However, there are two different compositions of bacterial cell walls, and they can be classified by a technique called a Gram stain. Gram-positive bacteria have a large amount of peptidoglycan and can therefore take up more of the stain. Under a microscope, it will appear a darker purple color. Gram-negative bacteria have less peptidoglycan and is sandwiched between the cell membrane and an outer membrane. These cells don't take up the stain as well, so under a microscope they will appear pink in color. How we can kill bacteria is through medications called antibiotics. One of the ways these medicines can kill bacteria is to act on the peptidoglycan and damage the cell walls. However, the gram-negative bacteria that don't have a lot of peptidoglycan are more likely to be resistant to antibiotics. Also, many bacteria have an additional sticky layer surrounding them. This is called a capsule. This allows bacteria to stick to each other or another substrate. Another external structure that allows bacteria to stick to other things are fimbriae. These are finger-like projections that act as Velcro to help the bacteria stick to other bacteria or to substrates. Some bacteria have the ability to be mobile, which is referred to as motility. Bacteria can move toward or away from certain stimuli. This is called taxis, when an organism is moving with a purpose versus just moving randomly. Remember, we studied taxis and kinesis on the second day of school when we did the pill bug lab. This is the same thing but with bacteria. Most of these bacteria get around through flagella, long hair-like structures that propel the cell. So that takes care of the outside of a bacterial cell. Now let's look at the internal structure. Again, prokaryotic cells are known for not having any membrane-bound organelles. However, certain bacteria can develop specialized membranes to compensate for not having certain organelles. For example, even though bacteria don't have mitochondria, membranes can specialize to be able to perform cellular respiration. Or membranes can act like the thylakoid membrane to perform the light reactions of photosynthesis, even though it doesn't contain any chloroplasts. Going back to the endosymbiotic theory, these could have been the precursor cells that were taken up and eventually became the modern mitochondria and chloroplast. But please know that there are current living bacteria that can do aerobic respiration and photosynthesis, just with these specialized membranes. Remember that all bacteria have DNA, but instead of multiple linear chromosomes like ours, they have one main circular chromosome. And instead of being housed in a nucleus, this chromosome is located in a region called the nucleoid region. It's just an area in the cell that's not surrounded by a membrane. Some bacteria also have separate DNA contained on smaller rings called plasmids. These plasmids are independent of the main chromosome and can replicate on their own. As we learned in our last unit, Bacteria can copy their DNA and make proteins through transcription and translation. However, there are differences between bacteria and our cells in these processes. We can exploit these differences and use antibiotics to target and kill bacteria cells while not harming ours. For example, while both types of cells have ribosomes, bacterial ribosomes are different than ours. So by targeting the bacterial ribosomes, we can stop them from making proteins that they need to survive, while our cells can keep doing what they need to do. Also remember that we've already learned that bacteria reproduce by a process called binary fission. One bacteria can copy its DNA and then split into two daughter cells. Since this is a form of asexual reproduction, you would think that bacteria would just be clones of each other, all with identical DNA. However, there's surprisingly a lot of genetic variation in bacteria. One reason for this is that they reproduce very rapidly. Bacteria can divide every 1 to 3 hours. And with reproduction happening very quickly, there's bound to be mutations occurring from inaccurate DNA replication. The other way we can get genetic variation in bacteria is through genetic recombination. This is combining DNA from two different sources. The three mechanisms of recombination you need to be familiar with is transformation, transduction, and conjugation. Transformation is when a bacterial cell takes up foreign DNA and incorporates it into its own. We've seen this process back in Chapter 16 in Griffith's experiment with the mice and the harmful and non-harmful pneumonia to prove that DNA is the genetic material. We'll be doing a bacterial transformation lab, and we're also going to look at this procedure more closely in Chapter 20 because it is a useful biotechnology procedure that has applications in research. Transduction is another way to recombine genes using viruses to bring DNA from one cell to another. Let's look at an example. Here we have a phage infecting a bacterial cell. A phage is a virus that specifically infects bacteria. The bacterial cell's DNA is broken up, and the virus does its thing to replicate itself. If you don't remember much about viral replication, I'll be putting up a virus video soon. While the viruses are assembling, some of the bacterial DNA accidentally gets packed into the head of the phage. And when the phage infects another cell, the bacterial DNA is injected into the cell. And this DNA can become incorporated as a part of the new cell. This new cell now has its own DNA and some DNA from another cell. This is genetic recombination. The last mechanism of genetic recombination is conjugation. This is when bacteria can transfer genetic material from one bacteria to another. However, this only happens in a one-way direction. There's a donor cell, and that cell gives DNA to the recipient cell. The DNA is moved between the bacteria through a connection called a sex pilus, or a mating bridge. So some people do refer to this method as bacterial sex. There are two methods of conjugation depending on a piece of DNA called the F factor, and F is going to stand for fertility for this purpose. This first method is when the F factor is in a plasmid, that small ring of DNA that's not the main chromosome. This is called the F plasmid. Here the top cell is the donor and the bottom cell is the recipient. A mating bridge forms between the cells. The donor cell makes a copy of the plasmid and passes it to the recipient cell. Both cells make sure the DNA is double-stranded, and now both cells have the F plasmid, and both are now considered donor cells. The second method is when the F factor is part of the main chromosome of the cell. In this case, the donor cell is referred to as an HFR cell. The F factor is transferred to the donor cell most of the time along with other genes that are shown in brown. Crossing over can happen to incorporate the new DNA into the recipient's chromosome, and any DNA that does not get incorporated into the cell will get degraded and broken down. Since this particular cell did not incorporate any of the F factor, it's still going to remain a recipient cell. The last thing I want to mention is the growing problem of antibiotic resistance. Resistance genes are found on plasmids called R plasmids. And like F plasmids, cells with R plasmids have the ability to create mating bridges and transfer these resistance genes to another bacteria. And that's going to increase the number of bacteria who become resistant to certain antibiotics. And that's where I'm going to end this video. This pretty much covers what you need to know about bacteria from chapter 27. Remember, it's just sections 1 and 2 from the chapter. So make sure you're keeping up with your workload. I hope you realize it gets a bit stressful and overwhelming when you fall behind. And we are going to be having a test soon, so let's kind of keep up with our work. And until next time, be a good human.